Okay, so yesterday we have seen, okay, the previous lesson we have seen, we have learned about the cyclic and non-cyclic photophosphorylations. So from the name here, photophosphorylations. So the name photo here means using the light energy. Phosphorylations basically means that the transfer of phosphate group, inorganic phosphate group. So basically means that we want to make or plant want to make this ATP. Okay, so let us look at this diagram. Okay? You look at this diagram. So this is the thylakoid membrane. Okay, can you see that? This is a thylakoid membrane. So these parts, they are stroma. Okay, the bridge color here is stroma. So this is a thylakoid space. The green color is a thylakoid space. So let us start with this photosystem 1. Can you see the photosystem 1? So yesterday, in the previous lesson, I told you guys that the electron won't jump up and down, but basically where the Z scheme of the electron flow indicates the energy level of the electrons. So if you look at this photosystem 1, when photosystem 1 receives the light energy, can you see that? The photon light energy. So this light energy strike on the pigments. So pigment absorb and then pass this energy to the reaction center. So photoexcitation of electrons take place. So this electron will be transferred along a series of electron carrier until they reach the NADP. So NADP together with the electrons and protons form this reduced NADP. So it means that photosystem 1 now lack of electrons, so that depends on photosystem 2. So the same thing, the light energy now hits on or absorbed by the pigment. So this pigment pass the energy, accessory pigment pass the energy to this reaction center P680. So when electrons, photos excitation electron take place, this electron again pass through a series of electron carrier. So against a different type of electron carrier. Okay, so now when this electron pass through this electron carrier, when they pass through this electron carrier, what happened here is energy is released. Okay, clear? Electron pass through, okay, or transfer along this electron carrier, energy is released. So when this energy is released, this energy is used to pump the proton from the stroma enter into the thylakoid space. From the stroma into the thylakoid space. Can you see that? So energy release, protons at the stroma is pumped into the thylakoid space. So therefore, it created a steep proton gradient. So it means that proton concentrations inside the thylakoid space is very high. Proton concentrations at the stroma is low. Can you see that? And this proton concentration is further increased by the photolysis process. Can you see that? Photolysis process giving out the proton as well. So it means that due to these two processes, the transfer of the protons from the stroma into the thylakoid space, as well as the protons released due to the photolysis of water molecule, therefore high concentration of proton inside the thylakoid space, low concentration of the proton at the stroma. So protons cannot pass through the phospholipid bilayer. They have to pass through this proton channel that associated with the ATP synthase. Can you see that? So the protons pass through the channel. So this channel associated ATP synthase. So when proton move from high concentration region to low concentration regions, they release the potential energy. So this potential energy is used to phosphorylate ADP to become ATP. So this ATP and reduced NADP will be used for Calvin cycle, which will be discussed later. Okay, clear? So recall back the ones we learned during the energy respiration. So you see that they are the same, chemiosmosis. So quick ones recapped here. First, electron pass through the electron transport change. energy is released. So this energy is used to pump the protons from the stroma into the thylakoid space. At the same time, photolysis of water releases the proton. So therefore, proton concentrations inside the thylakoid space is high, proton concentration at the stroma is low. So therefore, this steep proton gradients causes the protons to move from the thylakoid space into the stroma across this ion channel, which associated with the ATP synthase. So when protons move from the thylakoid space to the stroma, energy is released. So this energy is used to phosphorylate ADP so that the plants now can generate the ATP. So this ATP together with reduced NADP will be used in Calvin cycle. Clear? So a quick one. Energy is released as electron flow through a series of electron carrier. So proton now are pumped from stroma into the thylakoid space. So against the concentration gradient. So proton released from the photolysis further increase the concentration. And that we know the thylakoid membrane is impermeable to protons. So therefore, proton only can move out of the thylakoid space back to the stroma through facilitated diffusion, bifacilitated diffusion through this channel protein, which is associated with ATP synthase, and at the same time, energy is released. So with this ATP synthase catalyze the phosphorylation of ADP to ATP. So both processes, both cyclic and non-cyclic photophosphorylations will use this method. Okay, so therefore ATP is produced. So the process is termed as photophosphorylations. Okay. So with this, I've done for the chemiosmosis.